All right, so let's go ahead and continue. I had to take a quick break. So when we take a look at the body, we have in mitosis division of your somatic cells. And your somatic cells are those 23 pairs of homologous chromosomes. The chromosomes usually are paired together simply through genes that are carried at the same place. So the same characteristics, for example, attached earlobes, unattached earlobes, mid-digital hair, ability to curl your tongue, let's see, um, blue eyes, right? All of these are genes and characteristics that usually take place at the same place or locus. So the question is, if you were, let's say, born a female, right? Are all of your chromosomes fully homologous? And the answer is yes. If you were born a male, no, because male has an XY, but a female has an XX. So the only difference is simply because the male is an XY. Not all of your chromosomes are fully homologous when you're a male. They definitely are when you are a female. Okay, so if you take a look at chromosomes themselves, right? Here we have duplicate pairs of chromosomes. One here is duplicated, another one is duplicated, so we got duplicate pairs. You'll notice the banding pattern here. You got yellow, you got pink, you got green and blue venturing into yellow, and this turquoise color. You got yellow and yellow again and green. All these banding patterns are gene and loci that are traits that will carry certain characteristics. For example, this yellow one could be attached earlobes, right? This blue one down here could be for green eyes. This yellow one down here could be for, let's say, mid-digital hair, right? So anything that is a certain genetic sequence, it will actually be on a loci on the homologous chromosomes. And since they're paired up, they basically are paired up for chromosome 1, chromosome 2, 3, 4, all the 23 pairs we have. So just remember that they are paired chromosomes as opposed to just single ones. So each of the two sets of homologous chromosomes are called diploid. For example, your eggs and sperm, which are haploid cells, so your oocyte or egg and your sperm or spermatocyte, those are all individual 1N haploid cells. When fertilization occurs and the egg and sperm meet to create the zygote is diploid or 2N. Okay? Again, the zygote is diploid or 2N, but only when the egg and sperm meet. So really what you're taking a look at is a life cycle that is involving an alternation of both haploid and diploid cells, right? So, for example, over many, many generations, if gametes or sex cells were produced by mitosis, the resulting is the genetic material will start to divide over and over, right? If they were produced by mitosis, it would just be copies of one another. It wouldn't be through sexual reproduction. So your meiosis involves sexual reproduction or your gametes. Okay, so here we have the female, which obviously her gonads are the ovaries. Two pairs of those at the edges of the fallopian tubes, which are eventually connected to the uterus. The testes are external to the body simply because the sperm, which are produced inside the testes, this is just one singular testis, hence the IS. This needs to be external to the body because if the sperm were inside, internal, and the descent to the testes didn't happen, and you only had one external testis, then you'd only have to rely on the functioning sperm there. They are external because if they were internal, the sperm would all die because the heat of the temperature of the body would essentially kill the sperm. So that's why the testes are external to the body. Okay, so they both undergo meiosis, right? If egg and sperm meet, that egg and sperm essentially are haploid gametes. In the haploid stage, if they're meeting, fertilization happens, a fertilization membrane occurs, which prevents polyspermy and more than one sperm from essentially inseminating the egg. After this, you have your diploid zygote, which is a fertilized egg of a diploid 46 chromosome. This guy then is going to undergo mitosis, those normal phases of single division of prophase, metaphase, anaphase, and telophase. Okay, so in interphase, you usually have a pair of homologous chromosomes in a diploid parent cell that once they duplicate, they're going to create a pair, right? So you see one here, and one here. They're duplicating in interphase. This happened where? In the S phase of interphase. Where's the S phase? It's a synthesis phase between gap 1 and gap 2, which is all part of interphase. So again, interphase is very important. It's like 85% of your cell cycle, right? A huge chunk of that circle. So if that small piece of pie now involves prophase, metaphase, anaphase, telophase, and here we see double division of meiosis 1 and meiosis 2, you know you're talking about what? Just your gonads, right? Your sex cells of your ovary and your testis, right? Here we're starting to see, after the chromosomes duplicate, now you're starting to see in meiosis 1, the first division, those haploid cells will start to split, right? Those haploid cells of 1N and 1N will have duplicated chromosomes. Each of those will split again to have 1N, 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 or 1, 2, 3, 4, 4 haploid cells. So that's what happens at the end of meiosis 1 and meiosis 2. You basically have 4 individual haploid cells. Meiosis, unlike mitosis, is preceded by chromosomes duplication, but in meiosis, the cell divides twice, that double division I talked about. Mitosis is what? Single division. So it basically develops twice and divides twice to form 4 daughter cells. The first division of meiosis 1 starts with a pairing of homologous chromosomes, and then in prophase 1 of meiosis 1, also called crossing over, those homologous chromosomes exchange corresponding segments, the little legs cut off, and we talked about this earlier, which essentially means that you have an increased ability for genetic material as well as genetic diversity in the body. Meiosis 1 does what? It separates the members of each homologous pair and produces 2 daughter cells, each within one set of chromosomes. Meiosis 2 is essentially the same as mitosis, but in each of the cells, the sister chromatids of each chromosome separate. And again, in meiosis 2, which is the second division, you get what result? 4 haploid 1N cells. Checkpoint. A cell has a haploid number of chromosomes, but each chromosome has 2 chromatids. Your chromosomes are arranged singly at the center of the spindle. What is the meiotic stage? What is the meiotic stage? Whoops. Okay, so in this, it's going to be what? They're lined up, right? If they're lined up at the metaphase plate, they're in metaphase 2 because they line up 2 by 2 in metaphase 1. So these guys are arranged singly at the center, so they're definitely in metaphase 2. If they were lined up 2 by 2, they'd be in metaphase 1. All right, so we got meiosis 1, which is the first single division. Again, the most important part, the chromosomes will duplicate. Either this, you're starting to see the chromatids starting to condense. Now we're starting to see them separate. In prophase 1, what's going to happen? The spindle fibers will start to appear. The centrioles will migrate to opposite poles. In prophase 1, all the nuclear envelope disappears around the genetic information. In metaphase 1, you're starting to see that the chromosomes lined up at the metaphase or equatorial plate. The spindle fibers or aster bursts will start to appear from the centrioles. In anaphase 1, the sister chromatids will start to separate. And you also notice here in prophase 1 of meiosis 1, you're starting to see here, you see those crossing over right here? Didn't see it blown up, but the chromosomes you start to see are crossing over. Because they're crossing over in metaphase, when they separate, they're starting to separate their genetic material. So they're splitting. And so the important thing is also the spindle fibers will attach to the kinetochore of each of the centromeres to allow for the sister chromatids to migrate to opposite poles. So once they reach the poles, they're not in telophase. They're not in anaphase anymore. They are in telophase. So that's the next step. Okay, good. So you see the interphase as well as prophase a little bit better. Here we're starting to see the sister chromatids line up at the metaphase plate. You see that beautiful event called crossing over in prophase 1 of meiosis 1. Very important to remember. That's going to be a test question for sure. Where does crossing over occur in prophase 1 of meiosis 1. That's when your homologous chromosomes will separate simply because the chromosomes are lined up so closely to one another. You get this unique event. It doesn't happen in mitosis, only happens in meiosis 1, specifically prophase 1 of meiosis 1. And of course, we're starting to see that because they're crossing over, they're eventually going to split, right? But because they're crossing over, they're lined up at the metaphase plate in metaphase 1. In anaphase 1, once the sister chromatids separate, now you're starting to see that genetic information, right? It's completely split. You see red with blue, you see blue with red, and so on and so forth. These guys are essentially splitting their genetic material. You get what? Increased genetic variability, i.e. more diverse cells. So if we put the entire story together, right? After that, we're going to get two newly formed cells. Telophase 1 means the end. There's going to be a pinching in of the cleavage furrow. This is preceded by cytokinesis, which essentially splits the cell into two. If it splits into two, we got a second phase now of prophase, metaphase, anaphase, telophase. This is the second division where the same thing happens. The centrioles will start to migrate to opposite poles. The chromatin condenses. In metaphase 2, the chromosomes line up at the metaphase plate. Oh boy. In anaphase, oh boy, what happened? In anaphase 2, you're starting to see that the sister chromatids pull apart to opposite poles. And then in telophase 2, what do you notice? 1, 2, 3, 4 haploid daughter cells being created. Okay, so what happens at the end of two divisions of meiosis? After double division, how many cells do you have? Haploid, 4 daughter cells. Okay, good. So here we have plant cells, right? Lilies specifically, that you're starting to see here, them separated already, but you see this as well separated. So and this is almost between at anaphase to telophase. So they're flying apart from another. You see 1, 2, 3, 4 different cells. These guys are undergoing meiosis 2, and so you're eventually going to see 4 individual cells, haploid cells at the end of this. So both mitosis and meiosis begin with a diploid 2N parent. Their chromosomes will duplicate during the previous interphase. Mitosis produces 2 genetically identical diploid somatic daughter cells. Very important. Meiosis produces 4 genetically different, genetically unique haploid gametes. Why do we call them gametes? Because they're what? Your sex cells. They're your ovary and your sperm, right? So they're your ovum and your sperm, or your gametes. Okay, so we're going to take a look at mitosis and meiosis together, like back to back, and look at its similarities and differences. So one of the first things is you start off with your parent cell, and that's your diploid cell. These guys will eventually undergo duplication in interphase, which occurs in the S phase of interphase. After that, they will start to undergo prophase. In meiosis 1, it's the first division, and eventually the second division will happen. In mitosis, you just get one single division, right? So essentially in prophase 1, what happens? The centrioles migrate to opposite poles. The chromatin condenses and the nuclear envelope fragments. In metaphase, the chromosomes line up at the metaphase plate, okay? In metaphase 1, the pair of homologous chromosomes line up at the metaphase plate. In mitosis, for anaphase and telophase, the sister chromatids separate. So you start to see 2 cells. In meiosis 1, you start to see homologous chromosomes separate, and the sister chromatids remain attached. So what happens here is that you start to see in prophase 1 of meiosis 1, you see that site of crossing over between 2 homologous chromosomes. And so because that happens, you're going to see this very unique change in genetic diversity. The only reason why we have crossing over is to create what? If you think about it, 4 newly formed cells, right? If crossing over didn't happen, what would happen? You'd create 4 identical cells, but that's not what happens. In prophase 1 of meiosis 1, you get crossing over, which occurs in meiosis 1 to increase genetic variability. Okay, in the end, you're starting to see meiosis produce 1, 2, 3, 4 genetically different haploid cells. Okay, so again, in mitosis, you have 2 genetically similar, identical diploid cells. This is for what? Tissue repair, asexual reproduction, like budding, right? Which paramecium or amoeba or a yeast cell that eventually is used for beer, wine, and cheese production, right? Any one of your growth characteristics, like baker's yeast, is used for asexual reproduction. Those all undergo mitosis, okay? Growth is just what happens in the body, right? Normally, you keep growing the epithelial tissue that surrounds the skin, simply because the skin cells slough off, right? So because they slough off, they continuously have to undergo mitosis deep below in the dermis, which allows for it to go higher and higher and get to the epidermis, and eventually they're away from that bloodline, so they will slough off. So every day you're just sloughing off thousands of cells a day, which is kind of gross, but obviously if you don't take your shower, funguses can usually get into place. You get itchy, rashes, and all that stuff. Oil deposits, sebaceous and sebum. Oil deposits will start to get on the top surface of the skin. It's just going to become a gross, nasty bacterial breeding ground, right? Which is why, obviously, we have to take showers every day. That's why when we say growth, we're talking about mitotic growth. Tissue repair is going to be for what's inside the body. Let's say if my liver needs to repair, right? Undergo repair. Liver can obviously regrow. That's going to be tissue repair. If I have a bone that breaks, if I have a skin cut, if I have a deep gash or wound, if I have a complete or incomplete bone break, those are all tissue repair. What is that? Mitosis. You have two genetically identical diploid 2N cells. Your haploid cells here are just what again? Your gametes, your egg and your sperm, i.e. from your gonads, your ovaries, and your testes. Those undergo 4 different unique, genetically, totally different from one another. Why is that? Because of what event? Crossing over. Crossing over allows for, in prophase 1 of meiosis 1, to do what? Increase my genetic variability, so I get what? 4 totally different cells at the end of my double division of meiosis 1 and meiosis 2. Okay, so another thing we should take a look at is this thing called independent orientation, right? If my chromosomes in meiosis, which is basically used for sexual reproduction, and are randomly, and I got random fertilization, this will lead to totally varied offspring, which is why no brother or sister is the same, right? You have totally different siblings because of that event of prophase 1 of meiosis 1, which allows for independent assortment. Each chromosome of a homologous pair differs at many points, but random arrangements of chromosomes at metaphase 1 of meiosis leads to many different combinations of chromosomes in eggs and sperm. So random fertilization of eggs by sperms even increases this variation even more, because obviously it's not the same sperm that is obviously fertilizing the egg. Clearly through each ejaculation, each time you're trying for a baby, each unique event, there's one sperm that wins out, so each one is going to create a different type of kid. Okay, this is again why you have a variation. It says the total possible number of combinations produced by independent assortment in metaphase 1 is 2 to the 23rd, or 8,388,608 times. This number squared is more than 70 trillion. The authors rounded to 8 million and squared the estimate to 64 trillion possible combinations, but more precisely, the number of possible zygotes produced by a single pair of reproducing humans based solely on independent assortment and random fertilization is over 70 trillion. So you have 70 trillion different possibilities each time you get a kid, right? And you have a kid. There's 70 trillion different possibilities that this can have a unique sequence different from your other previous brother or sister. And obviously, if it's a brand new kid, 70 trillion different possible combinations. How amazing is that? That does what? It creates so much unique diversity. So there's no person that's the same on Earth. Everyone is unique. Everyone is special. Everyone's got a cool genetic background. Everyone's got a cool environmental growth. And so you can't really say, you know, you could base someone based upon their race, their education, their sex, their gender, their national origin, their creed, their color. None of that. Those are terms that are defined by us. We are all the same human species. The beauty is there's 70 trillion different combinations that each one of us could be a unique event, right? So this is what's so beautiful about biology. It's amazing how fertilization happens and how beautiful it really just takes, especially to create a very healthy infant. Okay, checkpoint. A particular species of worm, all right, has a diploid number of 10. How many chromosomal combinations are possible for gametes formed by meiosis? So this should be how many? So the diploid number is 10. You're actually going to get diploid 2n equals 10, right? So if you solve for that, that's 10 divided by 2, so n is 5. So it'll be 2 to the 5th power. 2 to the 5th power is 32. So that's how many different combinations are formed by meiosis. All right, so we got, let's say, a possibility. You got 2 equally probable arrangements of chromosomes at the metaphase plate. Possibility 1 looks like this arrangement, looks like this blue, red, blue, red. Possibility B is blue, red, red, blue. Okay, just swapped right here. So let's see what happens. In metaphase 2, you're basically going to see similarities where they're going to split. But here they're going to split blue and red and blue as opposed to blue and red, right? And this is a result of independent orientation, okay? Independent orientation. So the gametes are what? You could have same combinations, just 2 combinations of red and blue, or you could have a unique combination of 1, 2, 3, 4, right? So 4 totally different, actually 2 totally different and 2 totally different. So combination 3 and combination 4. And this is because of independent orientation. So let's take a look at this video.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes.
Nice.
In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is In sexually reproducing organisms, three processes lead to most genetic variation.
Uh-oh. Did I just mess that up? I think we're
Along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
All right, cool. That totally is amazing. That's actually pretty awesome. All right, so homologous chromosomes can definitely carry different versions of genes. The difference is that they can come from a totally different version of genes at unique corresponding points called loci. So normally what crossing over is, it's an exchange of information between non-sister chromatids. Remember, they're kind of standing next to each other, so their legs are just, it's like if two people standing next to each other and each person takes their leg out, right? That each person is a different person with obviously a different leg, or their own leg, right? So, but if it crosses over from, let's say, the knee down to the foot, right, or the bottom tail of that chromosome, basically what happens is you're going to have a different version of genes then. So typically, the example here is if each person has different genes, if we all have unique like genes, like blue genes, some are stonewashed, some have buttons instead of zippers, some have more pockets than others. The versions of genes are like different versions of genetic genes, representing options and sources of diversity. Okay, pretty cool example. Anyway, let's take a look at mice, right? My roommate actually starts, she was working at GW Lab, and all she does is ensure that the mice are still staying alive, right? Because there's so much information and research done with these guys. So if we take the Mus musculosus, that's our favorite genus and species name of the common household mouse, we take a look at its gene coat, its color, and it's brown. Or we take a look at it, it's white, right? And then we look at his eye color. This guy's black-eyed, brown coat, and he is pink, red-eyed. It looks almost red, right? Kind of scary rat, right? And then he's got the white eye. All rats are kind of scary. But anyway, some of them are really, really cute. But anyway, the brown and the black are homozygous dominant. So that's two capital C and an E. The white is a lowercase c and a lowercase e. Why does it remain C and E? Because that's the color of the coat, its coat and eye color. So the brown-eyed, black-eyed is like dominant for capital C and E. The white coat and the pink eye is lowercase for C and E. Okay, so these are the four different gametes that we're going to take a look at based upon coat color and eye color. So the first thing you notice is that you got the homologous chromosomes and you have this tetrad in a pair. So these homologous chromosomes are sitting next to each other. First thing you're going to identify is the chromosomes of each of the four gametes, right? We identified them, we separated them. These are the mouses we're taking a look at. Here is now what happens when it crosses over. If you actually look at the chromosomes, there is the crossover section called a chiasma. So chiasma is where the crossing over essentially happens over non-sister chromatids in prophase 1 of meiosis 1, a very important event which increases genetic variability. So this genetic of recombination, which results from crossing over during prophase 1 of meiosis 1, increases the variation even further. So checkpoint, if you were to examine a chromosome from one of your gametes, is it likely to look exactly like that chromosome from one of your skin cells? So it can't be. There's no way. So the answer is no, because each one looks like a different segment derived from the homologous chromosome, like a different loci. So there's no way that it's likely to look the exact same way. So if we take a look at and further increases of genetic variability, if they're paired up along with each other, your paternal and maternal chromosomes in blue and red, you see non-sister chromosomes forming that chiasma as they're crossing over just because they're standing next to each other, right? It's like intertwining someone's arms with and creating a link. We're basically creating genetic diversity as we do that, right? If let's say I cut off my arm and gave it to the other guy and we swapped arms, right? So this chiasma produces a hybrid chromosome. They start to then reassemble and recombine. If they recombine, the blue and the red start to split. You can start to see the separation of chiasma simply because this guy is going to increase genetic variability. Once they separate, though, in anaphase 1, those are the sister chromatids, each of them are going to contain a new segment from the original member of the pair. Therefore, in meiosis 2, you get 4 totally different, genetically different haploid chromosomes. Okay. All right, so there's those recombinant chromosomes. There's the original parental chromosome. So you get 2 recombinant chromosomes with new gene recombinations. All right, here's another good animation.
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
Okay, cool. I have to double check to see if this is recording, so hold on one second.